Gustation is the physiological term for taste, and obviously, taste is on your tongue, and more specifically, on your tongue, there are little raised bumps which we call papillae. Okay? These are sort of enlarged, raised portions of your tongue, and on those papillae is where we're going to find the taste buds. But there are different type of papillae that are present on your tongue. There are filiform papillae, right? And these are sort of filament shaped, so they're going to be sort of long and pointy like so. So they're filament shaped. Okay? You have foliate papillae, and these are sort of like leaf shaped. So think sort of like maple leaf, like so. Okay? You have fungiform, which, as you may imagine, are sort of mushroom shaped. So they will have sort of up and then sort of the mushroom cap, like so. And then you have what we call vallate papillae. And they're surrounded by a thick wall. So think sort of more trapezoid shaped, like so. Right? And the difference between fungiform and vallate can be a little bit difficult. Right? So think about the thick wall that surrounds that vallate. Now it's on these papillae where you are going to find sort of these taste buds, and a taste bud's going to sort of look like this rounded projection, and then it's just going to have a pore in it. And inside of here, you are going to have your cells, which are going to look sort of like this. And these are known as taste hair cells or gustatory hair cells. They have little projections on them, and those are going to have the receptors for the different tastings. And then inside of here, you're also going to have some supporting cells, which are known as sustenacular cells. And then we'll just label these green ones as our gustatory hair cell. Okay, so this all together is what makes up a taste bud. Okay, and so there are 5 different tastings or 5 different tastes that we talk about, right? And can you think of what those are? Salty, yep, everybody's going to get salty, sweet, sour. And can we get the other ones? Last 2 are the hard ones. You have bitter, and we have a savory taste, which is what we refer to as umami, right? And the odorant will bind, or not the odorant, the tastant will bind to the taste bud, and what causes the action potential in each of these different tastings is a little bit different. Okay, so salt, pretty simple. Salt, sodium chloride, sodium influx into the cell, right? So that one is pretty easy there, right? Sour is actually a proton, hydrogen ions that enter the cell, right? The sweet and bitter are a little bit different. They use a G protein linked signal transduction pathway, which we won't go into, right? And then umami uses the influx of amino acids, and we won't go into that process there, right? But there are the 5 tastings. Now, some people will say that these tastes are not equally distributed amongst the tongue. The taste buds that have receptors for these different tastings are unequally distributed about the tongue, right? So one fun way to sort of test that is to make some different solutions with these different tastings and then dip a Q-tip into the solutions and then dab it onto different regions of the tongue and see where you can taste those different things.